nucleotide sequence of DNA which determines the activities of cell or phenotype of an organism. But in 1940s, it was found that something else is also determining the phenotype of an organism. That was above the genetics. So Waddington in 1942, he coined a term for these phenotypic changes which were not due to the change in the sequence of DNA, but still they were causing a change in the phenotype. Such type of changes were named as epigenetics by Waddington in 1942. So the changes which are due to external modifications of DNA but without the change in the sequence of DNA were known as epigenetic changes. That means over and above genetic changes. So a consensus definition comes out to be a stably heritable phenotype which results from changes in chromosome without alterations in DNA sequence. And epigenome is a collection of all those chromatin patterns in cell, including both heritable as well as transient changes. Now the difference between genetics and epigenetic changes are, genetic changes we all know that they happen because of any mutation or any change in the sequence of DNA. These changes are generally inherited to the next generation. They may lead to mutations in a phenotype also. But epigenetic changes do not have any change in the sequence of DNA, still they cause variations in organisms. And those phenotypic changes are because of the external factors such as environmental factors, stress factors, behavioral patterns. Those kind of changes leads to epigenetic change. Genetic changes are mostly heritable. Epigenetic changes may or may not be heritable. There are different mechanisms of epigenetics. Now the epigenetic phenomena has been well established and three different mechanisms are well established which explains the phenomena of epigenetics. They are, first is DNA methylation. Methylation of DNA does not change the sequence of DNA, only external modification of DNA happens and that is an epigenetic change. The other change is modification in histone proteins. DNA is always wrapped in the form of histone proteins in chromatin. Those histone proteins are subjected to modifications and these are different types of modifications which happens to histone proteins like methylation, phosphorylation, acetylation, ubiquitination, sumoylation. That means these are small groups which are added to histone proteins after translation, that is post-translational modifications of histone proteins. They are also a type of epigenetic change. The third type of epigenetic change is also DNA methylation change, but that has been happening because of RNA. So that is known as RNA-directed DNA methylation. That means some RNA molecules are directing the sequences to be methylated. So it is known as RNA-directed DNA methylation. So I will talk about each of the mechanisms in brief, one by one. Before that, I will tell how these epigenetic mechanisms work. Since we know that there is no change in the sequence of DNA, still variation is there. So in terms of DNA methylation, what DNA methylation does is, it alters the affinity of proteins which binds to DNA. And we know that for expression of any gene, transcription has to happen on DNA. That means RNA has to be synthesized. For that, RNA polymerase and transcription factors and different activators have to bind on DNA so that transcription happens. But when the DNA is methylated, it changes the affinity of DNA to these transcription factors, to these activators, to the RNA polymerases. As a result, DNA is not accessible to RNA polymerases and different type of transcription factors. So gene cannot produce a RNA or a fully active RNA. So as a result, gene is inactivated. So that means gene is there is no change in the sequence of the gene, still the gene is not able to produce RNA or not able to express. The other type of modifications are histone modifications. Histone modifications reduce or alters the binding affinity of DNA in such a manner that when small groups like acetyl groups, methyl groups or different kind of groups bind to the histone proteins, histone proteins are already bound to DNA. DNA is negatively charged, these groups are positively charged, like proteins in the histone molecules like lysines, arginines, these kind of proteins are positively charged. So histone and DNA are tightly bound to each other. But when acetyl groups, methyl groups, these small groups are added to histones, they change the charge of histones. The positively charged histones gets reduction in their charge. As a result, their tight binding to the DNA is loosened. So histones are not as tightly bound to DNA now as they were bound earlier. So the loose DNA is now more accessible to transcription factors, RNA polymerases and other activators. So gene can now transcribe to produce RNA. So that means the binding of different histone molecules alters the affinity of DNA to transcription factors or RNA polymerases so that they can either bind to DNA or they cannot bind to DNA. So that produces a change in the expression of genes. So no change in the sequence of genes, only change in the expression of genes because of binding of histone proteins or because of methylation of DNA. So this is the basic of basics of how epigenetic changes alters the phenotypes of organisms. There could be different sources of epigenetic variations, like genetic variation arises. Similarly, epigenetic variations may also arise because of any spontaneous change, like we see in field sometimes mutation happens. So because of any spontaneous change, any epigenetic change in an organism may arise, or because of any genetic change in cis or trans. That means in genomes, generally transposable elements are present. So if any transposable element is activated and it moves into other regions of the gene, that may change other regions of the gene. So that is a cis change which may lead to epigenetic change. Similarly, trans change may also happen when some recombinations between different alleles of a gene may happen. That may lead to a trans change which could be genetic and which could be epigenetic also. Similarly, some white crosses may happen in nature that may give rise to any epigenetic phenotype. These are natural sources of variation. But epigenetic changes can be induced also. Induced sources of variation includes some mutations which we can create by treatment with certain chemicals like azacitidine, zebularine. Such kind of chemicals, when treated upon DNA, they may cause mutations in the DNA, not in the sequence, but in methylation patterns and histone modifications. When tissue culture plants are generated or some clonally propagated plants are produced, they may also sometimes lead to spontaneous changes in phenotypes that could be attributed to epigenetic changes also. Epigenomes are the genomes where two different kind of genomes vary in terms of DNA methylation patterns or histone modification patterns. Such epigenomes can be edited in artificially, just like genome editing is performed. Or mutations can be created through epirils. So these are some induced sources of variations for epigenetic changes. Then I'll talk about like what is DNA methylation. DNA methylation always happens at cytosine at fifth carbon position of this cytosine nucleotide. So it is normally called as methylated cytosine. When DNA methylation happens, cytosine molecules at their fifth carbon position methylates, that is a methyl group is added to cytosine so that 5-methyl cytosine is produced. This can happen anywhere where CG is present in the genome or where CNG is present in the genome. N could be any nucleotide except G, or where CNN is present in the genome, and this happens with help of certain enzymes which are known as methyltransferases. Now, when replication happens, the two strands of the DNA replicates in such a manner that one strand is original and one strand is newly formed. So, if the parent strand is having methylation in the strand, then the daughter strand which is produced will contain half methylation from parent and half of the strand is newly formed. So, that is why I have written maintenance methyltransferases. That means the newly formed half strand also has to again gain the methyl groups. So, maintenance of methylation. I will show you how it happens. These are the few enzymes which does this kind of DNA methylation. Methyltransferase MET1, that is an enzyme which is involved in maintenance of CG methylation. Chromomethylase is an enzyme which is involved in maintenance of CHG methylation. It is generally doing methylation at repeat regions, centromere regions and transposons. Then DRM, domains rearranged methyltransferases, they are generally doing de novo methylation. That means any DNA strand on which methyl group was earlier not there, newly added to that group, that is de novo. So, de novo methylation. And CMT2, that is involved in methylation at transposable elements sites. Here you can see that this was the original normal cytosine group. When methyltransferases enzyme transfer a methyl group to this cytosine, it becomes methyl cytosine. So here you can see that like there is no change. Cytosine earlier could form 3 bonds with guanine molecule. Here also when the cytosine is methylated, it forms the same 3 bonds with the guanine molecule, and in the DNA also it looks like the same. Only thing is that cytosine is methylated. So there is no change in the sequence, only the cytosine molecule gets methylated. What happens is like how this methylation affects the gene or how this methylation produces the variation can be seen here. Like these round structures are nucleosomes or histones around which this DNA is wrapped. So this is a chromatin. DNA is always present in the form of chromatin, like it is wrapped around nucleosomes in the cell. This DNA in methyl groups are converted to 5-methyl cytosine. That is, the methyltransferases are converting the cytosines to 5-methyl cytosine. You can see here this represent the 5-methyl cytosine. So when the DNA gets methylated, other proteins bind to it. These big ones are the other proteins, methyl binding proteins. After cytosine is methylated, these methyl binding proteins come and bind to the DNA. Binding of this helps in compacting of DNA. You can see the difference here. The DNA was very loose. All the nucleosomes were placed at a distance. But here when methylation has happened, all the nucleosomes have come close to each other. There is no distance. So the chromatin is compacted. That is the change which methylation brings. That is, whenever there is methylation of DNA, the chromatin which was actually loose, it gets compacted. Now you can imagine that this compact chromatin or compact DNA does not have any space where RNA polymerase can bind, where transcription factors can bind. So as a result, whatever genes are here, they are not able to express. No RNA will be produced, no expression of those genes would be happening. So as a result, the gene is same in unmethylated individual, gene is same in the methylated individual, but the expression is different. The phenotype would be different. So that kind of change is epigenetic change. Here you can see when it was normal cytosine, it is on, but when the methyl cytosine, the expression of gene is off. In this picture, maybe it is more clear. Like here you can see on the double strands of DNA, there are methyl groups, that is methyl cytosine. This is replicated. In replication, one strand retains the methyl group, one strand is newly formed. This newly formed strand has now two possibilities. One possibility is that when this strand replicates, both the strands do not contain methyl groups, half of this strand replicates, or this methyl group can be removed by demethylases. So this is the case here where demethylase enzyme has removed the methyl group, or on the second strand also this is again retained. So this strand methylase is added again. So all the three possibilities are shown in this figure where the two strands are containing the methyl groups. Then replication, while replication only one strand is having methyl, one strand is newly formed, then the possibility that demethylation may happen, methyl group is removed, or both the strands contain methylation, and this is the case where earlier there was no methylation but de novo methylation happened, that is newly added methyl groups add to the DNA. So this is how methylation, DNA methylation is happening. From this picture it is more clear, like you can see these black dots are methyl cytosine, that is methylated DNA. So when replication happens, one of this strand is same, one strand is newly formed. Here also one strand is same, one strand is newly formed. Then this is retained here, the second strand methyl groups are added by methyl transferases. Here also on the second strand methyl groups are added. So this is how methylation happens. Then I talk about the second type of modification, which is histone modification. The second type of epigenetic change happens in the chromatin, and we know well that chromatin is of two types, a euchromatin and heterochromatin. Euchromatin is generally active form of the chromatin, which mostly contains transcriptionally active genes, and most of the histone proteins here are hyperacetylated, that is they are more acetylated, because acetylation leads to activation of genes. Another type of chromatin is heterochromatin, which is highly compacted and it mostly contains hypoacetylated or less acetylated genes. It is inactive chromatin. This is just a representation of that chromatin, how it looks like. Chromatin is formed of histone and DNA. So you can see histone molecules H2A, H2B, H4, they form a tetramer, and two tetramers form an octamer. So a round octamer of histone around which this DNA is wrapped. Initially DNA looks like this. When it is formed, it wraps on nucleosomes, it looks like this. Then it gets compacted, it looks like this. And then scaffolds are formed when more compaction happens, and finally the chromosomes are formed. So in this individual nucleosome, you can see there are histone octamer. This is a histone octamer which consists of 8 histone proteins. These are amino acid tails, that means amino acids protruding from this histone octamer. These amino acids actually gets modified, which is known as post-translational modification. These amino acids modify by small groups, maybe acetyl group is added, methyl group is added, ubiquitin is added, phosphoryl is added. Like that, small modifications of these histones happens, and these modifications form the basis of epigenetic phenomena. Generally, the enzymes for these modifications, which are known as histone acetyl transferases, which causes the acetylation of histone. Histone deacetylases, they cause the deacetylation of histones. Histone methyl transferases, which adds methyl group to the histones. Similarly, histone demethylases, which removes methyl groups of the histones. These type of modifications and these enzymes are more well studied in comparison to other type of modifications. This is a picture which I have kept to show you. Like this is a chromatin in which nucleosomes are tightly packed to each other. These are N-terminal tails, amino acid tails, and these red are the small groups like acetyl groups, methyl groups, other kind of groups which are added to these histone proteins. Depending upon acetyl group is added, methyl group is added, these histone proteins are either compact or they are loose. If they are compact, no transcription will happen because RNA polymerase and transcription factors cannot bind to it. So as a result, this is a closed chromatin and which will suppress the function of gene. Gene will not be expressed and a different kind of phenotype will be produced. But some modification leads to open chromatin. Some acetyl groups are added, they lead to open chromatin. As a result, transcription can happen and genes can express. You can better see here, like this is a closed kind of chromatin and this is an open chromatin. When acetyl transferases, some methyl transferases happen, they make the chromatin open. So now RNA polymerase, different type of transcription factors can bind and gene transcription happens. But reverse is the situation happens when deacetylases or demethylases comes and they deacetylate the chromatin so that closed chromatin is produced. These are some common modifications you may see. K is a single amino acid letter for lysine amino acid. So the well-studied modifications of histones are H3, K9, K27, 29, K20. These are known to cause inactive chromatin or closed chromatin and they may lead to gene silencing. And modifications like H3, K4, acetylation, methylation, they are known to make open chromatin and as a result gene activation. Now this is the third type of epigenetic phenomena which I told is RNA directed DNA methylation. That means DNA methylation is happening but that is directed by RNA. These RNA are actually small RNAs. Small RNAs are produced in the cells when there is a double-stranded RNA produced. Some cells produce from their polymerase 4 a single-stranded RNA which gets copied into double-stranded RNA. That double-stranded RNA is cleaved into small RNAs which guides other proteins and molecules to do the methylation. These small round boxes are methylation at cytosine residues. So methylation of DNA is happening here, but because of small RNAs. Small RNAs are guiding some proteins to go to that location, bind to that DNA and do the methylation of DNA. So such kind of modification is RNA directed DNA modification. These are three different kinds of epigenetic modifications which are well known and well established. So in short, you can see like modifications to DNA due to DNA methylation, modification to histone proteins through post-translational modification of histones, or non-coding RNA or small RNA mediated DNA methylation or silencing. These are the three main pillars of epigenetics which can be shown over here. All these three pillars, they have an interplay among themselves, like small regulatory RNAs may lead to DNA methylation and small regulatory RNAs may lead to chromatin modifications also. DNA methylation may also interact with histones to do histone modifications, and histone modifications may also lead to DNA methylation and demethylation. So all the three phenomena of epigenetics may be interrelated to each other in certain cases. Now I move to the second part where I would explain the role of epigenetics in stress, particularly in biotic stress. This is a general model which shows all the three epigenetic phenomena like RNA mediated DNA methylation, hypermethylation or demethylation of DNA, and histone modifications when a stress, environmental stress or any type of biotic or abiotic stress is happening to plants. Any of the three phenomena may be active to produce a epigenetic change in plant. Like here you can see, when a DNA or an RNA virus attacks a plant, the RNA or DNA virus genome produces small RNA which are known as small interfering RNA or siRNA inside the plant genome, and those short RNA may lead to methylation of those viral DNA. As a result, now that viral DNA is methylated. Methylation in general means suppression of the gene or inactivation of the gene. So when viral DNA is methylated, it is not able to express that gene, not able to do transcription, not able to do proteins, produce proteins. As a result, virus attack is reduced, gene is inactivated, and virus resistance response is achieved by plant. Similarly, when any other pathogen or any abiotic stress is happening to the plant, plant undergoes either hypermethylation of its promoter regions. Hypermethylation means more methylation. More methylation means suppression or inactivation of gene. So, inactivation of genes, those genes which are helping the pathogen to survive in plant, to promote susceptibility, those genes may be hypermethylated and suppressed in the plants. So, that is a kind of epigenetic change, or demethylation or activation of genes which are involved in defense. That may help the plant in more expression of genes which are responding to defense of those pathogens. So, such kind of phenomena may happen. This could be more clear from this picture, where it is now well established that whenever plant is being invaded by a pathogen, a two-layer immunity system, PTI and ETI, that is pathogen triggered immunity or effector triggered immunity happens. This is now well established in case of plant immunity. What is it is? The first layer of immunity is PTI, that is pathogen triggered immunity. In this, whenever any pathogen, bacteria, nematode, aphid, fungus, any pathogen attacks the plant, this is the plant cell wall. This plant cell wall forms the first layer of immunity, because the plant cell wall is having certain receptors which we call as pattern recognition receptors or co-pattern recognition receptors. These receptors in the plant cell wall, they first recognize the signals produced by or the molecules released by pathogen as pathogen associated molecular patterns or microbes associated molecular patterns. Whenever any pathogen invades a plant, it releases some molecular patterns or molecules in order to attack that plant or in order to invade that plant. So, first layer of immunity is at the cell wall itself, where different receptors contains some molecules which interact with those molecules released by the pathogen. So, as a result, some downstream signal is passed on to the nucleus of the cell. A number of genes are involved, so that plant gets ready to respond to the attack of pathogen. A number of genes in the plant, the defense genes in the plant, gets activated so that plant can respond to the invasion of pathogen. That is the first layer of immunity. Even if somehow the pathogen is able to invade or to break this first layer of immunity, that is at the cell wall, and is able to form a hysteria or is able to secrete some proteins which are known as effector proteins into the plant, then the second layer of immunity, that is ETI, that starts working. In that ETI, plant has certain genes which are known as R genes or resistance genes. Those resistance genes encodes NBS LRR kind of proteins mostly, in most of the R gene cases. Those NBS LRR proteins are commonly known as NLRs. We call them NBS LRR like receptors or proteins. Here it is represented by NLRs. Those NLR proteins interacts with the effector proteins produced by the pathogen. As a result of interaction, they produce a downstream signal, passes it on to the cell nucleus, so that cell nucleus now gets ready to respond to the attack of pathogen in which pathogen has secreted its effector inside the cell. As a result, these NBS LRR proteins starts killing the cell of the pathogen, which is known as a hypersensitive response, in which the localized cell death in that region happens, and it starts transmitting the signal to the nucleus, so that other downstream genes which are involved in defense, like respiratory burst diseases, peroxidases, oxidoreductases, other defense responsive genes, disease responsive genes, all those genes gets activated, so that now plant is ready to respond to the infection by pathogen. So, this is the second layer of immunity, ETI. Now, epigenetic plays a role here, like whenever PTI or whenever ETI kind of immunity happens or response happens in plants, a number of genes are involved in the plant which gets activated or which gets suppressed, which were leading to susceptibility, they get suppressed, and as a result plant is able to respond to the pathogen. The activation or suppression of these genes is controlled by various epigenetic factors, like some of the genes may be suppressed because of DNA methylations, some of the genes may be acted because of histone modifications, which we do not know till now. We know this two-layered plant immunity system is working, but we do not know that the differential expression of those genes may be because of certain epigenetic controls also. Till now we were looking only towards the genetic control of those genes, but there could be some epigenetic controls of those genes which are giving the resistance response to plants. Like if you see, if pathogen has induced either hypomethylation or hypermethylation of genomes, what could they do? This figure shows this is the gene region, red one is gene, black one is promoter, and blue one is a transposable element which has been inserted into the promoter. So if methylation, hypermethylation of any of these regions happens, they may lead to inactivation of gene. So if suppose this was a gene which was responsible for promoting, enhancing susceptibility to plant, then hypermethylation of such a gene region or a promoter region will lead to suppression of that particular gene, and as a result plant may get the resistance response. Or hypermethylation of hypomethylation of a defense responsive gene. Suppose it was a defense responsive gene, less methylation or hypomethylation of that gene means more activation of that gene. So that defense responsive gene would be more activated. So in general summarization which I have found from literature is that generally bacterial and fungal phytopathogens induce hypomethylation of defense and stress related genes, means they activate more the defense and stress related genes, whereas cyst nematodes mostly activate hormonal and developmental process related genes. That means they do less methylation of these kind of genes. In case of biotropic and necrotropic fungi, different kind of methylation patterns have been found in literatures. And in case of viral infection, the methylation patterns of host have not yet very well characterized. So there are no such examples in case of viral infections where host methylome could be characterized. This figure shows that what could this methylation be doing in the host, like if it is induced by pathogen. Pathogen induced DNA methylation may either lead to transcriptional reprogramming or it may change the stability of the genome. That means that if methylation happens in gene body regions or in promoter regions or in transposons, they may lead to different kind of RNA that could be functional or that could be dysfunctional, because when methylation of transposon happens, transposons are suppressed. Hypermethylation of transposons means transposons are suppressed. They will not move into different genes. So as a result, defense response genes would be activated and they would give defense. But if less methylation of transposons means they would be active, they will move into defense genes and suppress the defense genes. As a result, susceptibility or susceptible phenotype may produce. Similarly, the methylated DNA is more prone to mutation because methylated DNA may get deaminated. As a result, nucleotides may change. Cytosine may change to thymine, guanine may change to adenine. So as a result of changes, some spontaneous mutation may happen. Transposons may move from one location to other location. If they are active, they are less methylated, they are active, they will move from one and lead to genome instability. Similarly, some genome rearrangements like non-homologous joining may happen. Something may happen which may lead to instability of the genome. So in general, pathogen-induced DNA methylation may lead to either transcriptional reprogramming or instability of the genome, which will add to the phenotypic and genetic diversity of plants. In order to show this, like what epigenetic change can produce, variations can be produced, I have taken one case study for leaf rust resistance gene. This is part of our collaborative work which we are doing with CCS University Meerut. This is with respect to gene LR28. There are a number of leaf rust resistance genes. We have taken one gene LR28. For this gene, we have near isogenic lines in the background of HT2329. One is a susceptible line, other is near isogenic line, which differs only for this leaf rust resistance gene. S here is the susceptible genotype, R here is the resistant genotype carrying the gene LR28. So from this figure you can see, like this figure represents the changes in DNA methylation patterns of susceptible and resistant genotype. From here, this you can see this is S96 versus R96, means the susceptible genotype after 96 hours of inoculation with pathogen, leaf rust pathogen, versus the resistance genotype after 96 hours of inoculation. Here you can see more number of regions are hypermethylated, that is more methylated in case of resistant genotype, and in case of susceptible genotype less number of regions are methylated. So that from this study we got the idea that maybe more number of genes are activated during susceptibility, and later on during resistance those genes are suppressed, and a different set of genes are activated during resistance, but they are less in number. More number of genes, we could conclude that the genes which were differentially methylated, they were playing either directly a role in disease resistance, or maybe indirectly they were related to disease resistance or susceptibility. Methylation was observed in all regions like promoters, exons, introns. A large number of methylated regions were involved in maintaining transposable elements, that is transposable elements were kept suppressed so that they do not move inside the genome, do not make the genome unstable. So a large number of transposable elements were suppressed, and then it was found that DNA methylation was not always related to inactivation of gene, because wherever we found DNA methylation, we do not found all those genes suppressed. So that means that in addition to this DNA methylation, other epigenetic factors like histone modifications, small RNA directed DNA methylation, they might also be playing a role in resistance to leaf rust. So we could find out that maybe in addition to genetic controls, the resistance to disease by pathogens might also be regulated by certain epigenetic controls. At least partly it could be epigenetic controls also. These are a few, this slide shows a few genes, these genes which are actually known to play a role in defense or resistance, and they were found to be differentially methylated in response to leaf rust resistance, like CLP protease. It is already known in case of fungal, bacterial and insect infections. Glutathione transferases, they are already known in case of salinity and other oxidative stress. Terpene synthase, this is already known in case of fusarium crown rot. Similarly, these transcription factors, they are already known in case of powdery mildew and leaf rust infection. So that means these genes were already known for producing response to resistance response to different kind of pathogens and abiotic stresses, but it was not known whether they could be epigenetically regulated or not. From our study, we found that these genes were differentially methylated also. So that means there could be a bigger role of these epigenetic controls in differential expression of these genes, which we still do not know. Then we also tried to find out certain histone modifications in case of that leaf rust resistance gene. We selected these two histone modifications, H3K4 and H3K9. Both of these were selected for acetylation, that is activation of genes. We selected these 6 genes for studying these histone modification. These 6 genes, their functions were already known in different crops for their response to disease resistance and abiotic stress resistance. And the purpose of choosing these 6 genes was that these 6 genes were differentially expressed in response to leaf rust resistance. So we wanted to see whether the differential expression of these genes could also be regulated by histone modifications or not. So that is why we selected these 6 genes. This is the region of promoter. P1 is proximal promoter, P2 is distal promoter region of these 6 genes. Because any change in the promoter region of those genes, any modification means it will not lead the expression of those genes, because transcription factors cannot bind to the promoters and expression of gene would be altered. So we chose the promoter regions of these 2 genes. This shows the sequences or the motifs which were responsive to defense, which were already known to play a role in disease resistance or defense. That is why these 6 genes were chosen, because they have already certain sequences in their promoter regions which binds to the transcription factors in response to disease. This shows the result. Maybe I can summarize this. Y shows yes, Y represents yes, N represents no, and NC means not calculated. This is susceptible genotype, this is resistant genotype, this is susceptible, this is resistant. This is proximal promoter, this is distal promoter. So for all these genes where Y is obtained, that means their histone modification was obtained. That means that acetylation of histone was there in that region wherever there is Y. Wherever there is N means there was no histone modification observed in that region. Then we see that this histone modification is related to gene expression or not. So the conclusion was that only 2 genes, N-acetyl transferase gene, showed higher expression in susceptible Neelu, and its deacetylation showed lower expression. Similarly, peroxidase also showed higher expression. So that gave us the conclusion the genes which were differentially expressed could also be regulated by histone modifications also. At least 2 of these 6 genes were found to be regulated by different histone modifications which were leading to activation of those genes. So from these results, we got an idea that maybe in case of leaf rust resistance, whatever differential response we are getting, some of those genes, at least part of those genes, may be epigenetically governed also. These were the only studies which I could get for fungal infection in wheat in case of leaf rust, because not much work has been done in respect of DNA methylation and histone modifications for any kind of resistance, rust resistance in wheat. In addition to that, I got some more studies related to other fungal pathogens, that is powdery mildew in wheat. There were 2-3 more studies which indicates the role of DNA methylation in some genes like receptor kinases, peroxidases, pathogenesis related genes. Those kind of genes were found to be methylated and active in case of resistant genotypes in powdery mildew also. So from these studies, growing evidence is coming that maybe some of the fungal diseases in wheat might be, some genes might be epigenetically governed also. Then I got one study, a recent study where this epigenetic variation was studied on pathogen also. It was found that the effector genes of pathogens, they may also be methylated in, and the variation in the response of these effector proteins may be due to methylation in the genes of those effector proteins. So that was also studied in one case. In case of Puccinia graminis, it was found that the effector gene was enriched for methylation in transposable element region by small RNAs, that is RNA directed DNA methylation was observed in case of Puccinia graminis in a recent study. This slide I have put to just emphasize that since epigenetics is a growing field And a number of studies, though you can see that there are something around 8,000 studies which have been going in the field of epigenetics for all kind of pathogens, for all kind of stresses, for different kind of organisms. But those which relates to the microbes, that is the green one, they are only 1,000, which relates to the role of epigenetics in respect to host pathogen interactions. They relates to microbes. They are approximately 1,000 in all crops, in all type of pathogens. And among them, those which relates to fungal microbes and the epigenetic control of them in host pathogen interactions are only 21 till now in all different crops, including all different fungal pathogens. So there is much more that needs to be learned about the role of epigenetics in host pathogen interactions, including wheat and all other crops with respect to these epigenetic controls. This slide I have put to just emphasize that we are only thinking about the role of epigenetic control with respect to the host, like what changes are done in the host or what epigenetic variations occurs in the host when host pathogen interaction is there. But there could be certain epigenetic changes in the pathogen also when host pathogen interactions are there. So I tried to find out whether there are any reports available for rust fungi. There were none, but there were some reports available for Magna Porte oryzae, a fungus, in which epigenetic regulation controls have been studied. So they found that all three, DNA methylation, histone modifications and small RNA, all three type of epigenetic modifications were reported in this fungi, Magna Porte fungi. But the questions which remain to be answered here were like whether any DNA methylations which were happening may lead to a role in evolutionary, may have a role in evolution of that fungi, or whether any histone modifications, they are leading to more adaptive evolution of that fungi, or maybe there is a host pathogen co-evolution which is happening because of these epigenetic changes in the fungi. Some reports says that pathogen genes which are responsible for producing effector proteins, those genes are also epigenetically controlled, because effector proteins are not needed to be produced all the time by the pathogen. Pathogen only needs to produce its effector protein when it has to invade a host. So why will it waste its energy every time in producing those pathogen genes, those effector genes? So pathogen might also evolve some mechanism that it will produce its effector gene, it will express its gene only when it is invading the host. So that could be one reason, like pathogen might be adopting epigenetic mechanisms for its growth and development, for maintaining its growth and development at a time when no more effector genes or nothing is required. And later on, when effector genes are required for invading pathogen, maybe some other epigenetic controls are activating the genes and effector proteins are produced. That is one speculation that could be there. Or maybe for evolution of pathogen, since host is continuously evolving to make it resistant to the different kind of pathogens, pathogen also needs to evolve themselves in order to attack the host. So maybe from that adaptive point of evolution, pathogen might be doing some histone modifications or some epigenetic changes. That needs to be still answered, that is not known. And another thing is that there are also known certain secondary metabolites. Those secondary metabolites are also inducing the pathogen to do some epigenetic changes. So could it be something new which is still now not known, that those pathogens could be induced by those metabolites to express some genes or to suppress some genes? So these were some of the questions which still remains to be answered when we look it from the pathogen point of view, when we look at the epigenetic controls from pathogen point of view. Then comes another question which relates to the inheritance of these epigenetic changes. This slide I have put to summarize, like whenever there is an abiotic or biotic stress in any natural population, any kind of epigenetic change may happen that may lead to alteration in expression of gene. Now, if that alteration of expression is transient, is not required later on, it is reversible. All epigenetic changes are reversible. Whenever they are not needed, whenever the organism is not under stress, any kind of stress, it has adapted, then they may reverse and it is a short-term change or a transient change. But these changes may be heritable also. If they are mitotically inherited, that is, the changes are there inherited only in one generation, that particular individual has that change which has suffered or which is prone to stress, then that change is within that generation, it is mitotically inherited. If that change passes on to the next generation, it is meiotically inherited. So till now, certain changes, certain epigenetic changes are found to be mitotically inherited, and few of them have been found to be meiotically inherited, especially in case of abiotic stress tolerance. I have not come across any epigenetic change which is meiotically inherited in case of biotic stress. This is something which tells about stress memory. Like whenever any pathogen attacks a plant or whenever any stress is there to the plant has a memory, it undergoes certain epigenetic changes. That epigenetic changes remains in the memory of the plant. So when the plant is attacked second time or when the plant is invaded second time, that stress memory will help the plant to regain those epigenetic changes, and then plant will respond in a better way to those changes, to those stresses, biotic or abiotic stresses. That is known as stress memory. That stress memory could be transgenerational or intergenerational. Intergenerational means if that stress has happened to the plant and that has been inherited in the next generation, in the first next generation, that is intergeneration. When that is inherited up to next generation, that is 2 stress-free generations, it is transgenerational, means inherited across generations. So that is a transgenerational memory. A plausible explanation for this stress memory could be like whenever replication happens, some of the nucleosomes are retained from the original parent and some nucleosomes are rebuilt. And since nucleosomes contain histones which undergo modifications, so that means some histone modifications are there already inherited from the parent at the time of replication. So that could be one reason for retaining the stress memory of plant or any organism due to this epigenetic change. So now I come to the conclusion of this, like what could be the future implications for these epigenetic changes are, these DNA methylation patterns represents a very important regulatory layer of plant immunity. So they can be exploited more in order to improve the disease resistance in plant. We know something about DNA methylation, but not that much in detail with relation to different diseases in plants. So if we identify any pathogen effector proteins, then we may know what this pathogen effector protein is producing change in the host, what epigenetic change has happened in the host with respect to this effector protein. Since we do not know effectors for many of the diseases, we are not able to study the epigenetic changes which are happening because of these effectors. So identifying those pathogen effectors may directly or indirectly help us in understanding the DNA methylation patterns in a more better way. Stress priming which leads to retaining of stress memory. Priming means like exposure of something to something. So if a plant is exposed to biotic or abiotic stress, that is a stress priming. A little bit of exposure to that stress will lead the plant to respond in a better way later on when the plant is again exposed to that stress, because it will have a memory of that stress, that is stress memory. So stress priming may lead to a stress memory which could later on help in better response of that plant. Epigenetic diversity may be created if we know the different epigenetic phenomena that may lead to better crop improvement possibilities. And it is also necessary to study the different epigenetic mechanisms in order to know the stress tolerance of plants with respect to the adaptation, with respect to the evolution, and with respect to the diversity of the plants for different stresses, biotic as well as abiotic stresses. That is all about epigenetics and its role. Thank you. Any questions?
Which compound donates the methyl and acetyl group in this methylation and acetylation reactions?
I couldn't get you. I'll come down. Which type of?
Which compound donates the methyl and acetyl group in this methylation and acetylation?
S-adenosyl-methionine. That is a compound which donates the methyl groups, which is transferred by DNA methyl transferases to the cytosine molecules.
How will we detect the methylation and acetylation in DNA?
There are different techniques to detect methylation, and I was about to include like methylation sensitive amplification polymorphism. This is one technique. Another technique is bisulfite sequencing. In bisulfite sequencing, when the DNA molecule is exposed to bisulfite, sodium bisulfite chemical, then some of the cytosines which are methylated, they will remain as such, but those which are unmethylated, they will get converted to U, that is uracil. So then you can differentiate the methylated cytosines and the unmethylated cytosines. If you take a control, compare it with the untreated bisulfite, untreated and the bisulfite treated. So you will be able to compare the cytosines.
And there is one more thing. Can we target the specific region of the chromatin for methylation or the acetylation?
You can target specific region of chromatin, means specific genes you want to target. Yes. For histone modifications, definitely, because histone modifications you will do with using antibodies for those kind of histone proteins. Whenever you have to identify the modifications in histones, you always have to use antibodies. So if you want to target acetylation, you will have to use an antibody for histone acetylation. If you want to target methylation, then you have to use an antibody which is specific for methylation. In case of DNA methylations, some specific regions you can target with the help of PCRs if you want to target a particular gene. So if you use a methylation sensitive PCR for that particular region, you design primers for that particular region, then maybe you can target a smaller region also. That also you can do. Sumoylation. Sumo is a protein group, a big protein like ubiquitins are there, they are also proteins. Like that, sumo is also a protein group, a big protein. Addition of that sumo group to any protein is known as sumoylation.
Why you focused on cytosine methylation? Why you didn't go for adenosine?
Whatever methylation is known or which leads to epigenetic change is because of cytosine, because that methylation doesn't produce any change in the sequence. Other kind of methylations could be altered, like demethylations may be happened in those, so they are not stable changes. So that is why.
Thank you, ma'am, for informative presentation. So, in one of the slide you said basically two types of methylation chemical like ICAR-IARI, hypomethylation and hypermethylation. So, in case of different pathogens, you talked about the biotrophic pathogens, rust and etc. So, in that you mentioned that there are different opposite patterns of methylation can exist. So, what are these?
Opposite patterns of methylation means generally we believe that if hypermethylation is there, it will lead to gene inactivation. So if that was a pattern which is observed in case of biotropic pathogen, that was not observed in case of necrotropic pathogen. Means if hypermethylation is there, then some of the genes were not inactivated, some of the genes were found to be activated. So that is what I mean by opposite patterns of methylation. Like some genes which needs to be, which should be inactivated as per our knowledge of hypermethylation was not there. So that is what opposite patterns we could get.
So in that case, the particular phenomenon does not exist in case of
Then we believe that maybe the change is not because of methylation. Maybe some other factors, some genetic factor or some other epigenetic factor might be playing a role. But that we cannot attribute to DNA methylation because the pattern is not the same. So we are not knowing. Okay, thank you everyone for your patient hearing. My slides I have taken.
Thank you, ma'am, for giving such nice lecture.
Okay, thank you. Huh? Thank you. Namaste ji.